Good morning, everybody. First, I'd like to thank Ion and the people organizing this meeting for the kind invitation to present today. We switched gears a little bit, so I'm talking about AML or leukemia. Today, I will talk about two projects which we have done, both together with lonTorrent, one with major help from lonTorrent, and the other one mostly on our own initiative. And just to give you a broad background about AML, AML is a malignant disease of hematopoietic stem cells. It is quite rare, affecting about 1.3 individuals per 100,000 in developed countries. The median age of patients is in the range of 60, so it's a disease of the elderly. The hallmark is a massive expansion of myeloid blasts in the bone marrow, which then confer all the problems with which these patients have, namely that they are bleeding, they are losing their leukocytes, so we get infections. Diagnosis involves, of course, morphology. The microscope is still the most important part in the diagnosis of AML. But in the meanwhile, immunophenotyping and cytogenetic classification has become especially important, and also molecular tests have been employed. The treatment of this disease still relies mostly on chemotherapy, although some targeted approaches have been developed in the past. They are restricted to certain selected subgroups. For example, APL can now be cured in selected patients without chemotherapy. But in the majority of the patients, especially in the high-risk population, hematopoietic stem cell transplantation is still the method of choice to achieve long-term cure. Smart people have deployed at that time, 40 years ago, for the first time, the most comprehensive analysis method at that time, that was metaphase, cytogenetics, and G-banding. And they were able to sub-classify AML mostly in two groups of patients. We have on the one side patients with cytogenetic abnormalities, and these are mostly reciprocal translocations, monosomies of certain chromosomes or trisomies, and deletions of major parts of chromosomes, or even a complex karyotype as depicted on this slide. On the other side, half of the patients do not show any cytogenetic abnormalities, so have a normal karyotype. And especially in this group of patients, we have made major progress in understanding the mechanisms of disease. With cytogenetic classification, we were able to sub-stratify the prognosis of patients into three major groups, namely the good risk population characterized by the CBF leukemias and the T1517 translocation, the APL. On the other side of the coin, there is the high-risk population, mostly characterized by the complex karyotype and the monosomal, so-called monosomal karyotype. And in between, there is all the rest, mostly the normal karyotype population. With the more comprehensive analysis method of next-generation sequencing, we meanwhile learned that we have a number of abnormalities in AML. And AML, we are on the lucky side. AML is actually a disease which is quite genetically stable compared, for example, to many solid tumors, which have at least 10 to 100 times more alterations per megabase. But still, when we only analyze a certain group of genes, which has been done already in the past, we see how complex this disease is in sub-stratifying on the molecular basis. So we can identify certain groups of patients having alterations in genes like, for example, NPM1 or CBP alpha. This got even more complex, the landscape of AML, with the introduction, which was recently published by Timothy Lay's group from the University of Washington, within the Cancer Genome Atlas Consortium. This group sequenced 200 patients with AML, either by whole exome or whole genome sequencing, and they could identify 1,600 genes with mutations, and 260 of these genes were mutated at least twice. A typical AML in this analysis has between 12 to 15 mutations in genes, and we could identify 4 to 5 recurrent changes per individual. This information is already within the clinic, so we are using it in a clinical scale. The WHO, within the most recent release of the Blue Book, included alterations like nucleophosmin 1 mutations or CBP alpha mutations as disease-defining if they are present within a certain blast range. So well, the presence of these mutations defines a disease as AML. And groups have defined, based on certain cytogenetic as well as molecular alterations, risk groups, prognostic classifications, and this can be redefined using mutations like NPM1, FLT3, CBP alpha, and others. And you can see clearly that we are able now to further subdivide the group of patients with different prognosis. And this has been confirmed by several groups worldwide. So there is a high need to make genetic information available on a large scale. And this was a point where we teamed up with lonTorrent to build a research panel, a network to identify and to define a research panel to test whether it's possible to have a comprehensive, integrated tool for analyzing patients with AML on the molecular basis. We met in London in June, and these were the groups which were involved, and together with the Falcon team from lonTorrent, and we sat together to define a list of genes which might be mostly appropriate within such a panel. And the reasons to integrate a gene in the panel were either the high prevalence, so genes which were very common in AML, genes which were disease-defining, like CBP alpha or nucleophosmin, or other genes which are associated with particular poor or good prognosis, like, for example, TP53 mutations, which are really a poor prognostic factor, or other genes which are potentially usable to identify targetable genes or proteins, like, for example, c-KIT or Sybil or other genes, which might be useful for drug treatment later on. So we ended up with a panel of 19 genes which are included in the first version, and this is so-called core panel. And it's clearly thought that it might be later extended to have a final list of up to 30 genes which are currently tested in these patients with AML. And we, for reasons of costs and effectiveness, combined exon coverage of the genes with hotspot coverage in those genes which are only mutated in a certain region of the gene. We have a two-pool design currently with 337 PCR primer pairs. So per pool, 170-plex reaction. Median size is around 200 base pairs, 180 base pairs. And we only need 20 nanograms input per entire reaction, so 10 nanograms per pool. We tested very recently, only a month ago, we sent the samples to lonTorrent in California. This was a test set of 21 research samples which had been characterized by us using either Sanger sequencing or different technologies like fragment analysis or others. And the people at lonTorrent used their most recent releases of the different diagnostic or research tools they have in hand, the AmpliSeq 2.0 library, the 200 kit, and the PGM sequencing kit version 2. And they put 4 barcoded samples per 318 chip. And you can see clearly that the coverage we achieve is much higher than probably needed at the final end. So we will probably end up with a sample number of 6 to 8 samples per chip. 95% of the target base coverage was above 500-fold, which is much higher than probably needed to achieve a sensitivity of 5 to 10%. Data analysis involved the Torrent Suite software 4.0 and the most recent, not for everyone available, release of the lonTorrent reporter software. This slide depicts the results achieved. We have a fairly uniform coverage for all amplicons, above 90% for almost all samples. We have also a very decent percent of reads on target. There is almost no strand bias in the majority of the amplicons. And you can see also that the majority of the samples have a very high coverage. So this slide is not meant to be read in detail. So what you can see here, green depicts that the mutation which was tested was really detected. And you can see that for the 21 mutations which we tested, 20 were detected either directly by the software or with small adaptions. Only one mutation in the A6L1 gene was not directly detected. These are just some screenshots of the iReporter software, which appears to be a quite useful tool to do the analysis. This is the first analysis of such a sample, which we sent to lonTorrent. You see that there were 32 alterations present in this sample. And this also includes, of course, the normal germline. And by applying certain filter algorithms, you're able to pull that down to 5 variants by just extracting the germline variants and annotating it for the cosmic and other databases. And you can see clearly that you end up with those mutations which were really present in the sample, which we knew already. So this software appears to be a quite useful and applicable tool, and important tool also to make this usable on a clinical scale. So this is the 2 mutations which we did not directly identify. One was clearly seen in the analysis, but was by the torrent suite, but was not called by iReporter. And there is clear evidence that this is due to an algorithm change or bug, which will be cleared very soon. And the second was probably due to mispriming of overlapping amplicons, which induced this A6L1 variant not to be detected. So in summary, it appears that even this was only a first shot, a very first shot. The results are already quite convincing and quite encouraging. We have a very good and uniform coverage and a high specificity with reads on target. The coverage is totally sufficient and can be even decreased. And we called almost all of the mutations present in the sample. So the next steps will be to redesign the panel, and there will be some adoptions to have better coverage in some subgroups. And there are some genes included which have very problematic GC content, up to 80%, for example, for CBP alpha, which will be challenging, but we are quite confident to solve this. And also the long indels can be probably managed. So we expect to have a alpha beta version of the panel ready late Q4, probably early Q1, and this will be then distributed to interested groups later on. So this brings me to the second part of my presentation, where I want to focus on an application which we are interested in, which is called minimal residual disease. When you have a patient with AML which enters the clinic, this patient typically has about 10 times 10 to the 11 leukemic cells in his body, summing up to almost 1 kilogram of leukemic burden. If you have a very effective treatment, you can kill probably 10 to the 6 or 10 to the 7 leukemic cells, but you see that still some leukemic cells will survive the treatment. Some patients with ongoing treatment will be finally completely cured of their disease, and these patients definitely do not need additional treatment. But we know from our clinical experience that the majority of patients will eventually relapse earlier or later. And we also know from our experience that detection of this relapse early would help us to stratify treatment, new treatment, and help this improving survival in these patients. And we have a number of tools, molecular tools, to detect disease more sensitively than morphology, which has only a very limited sensitivity of 5% by definition. FISH is a little bit more sensitive at a 1% level. Standard PCR probably at the 10 to the minus 4 level. qPCR, nested PCR can detect 10 to the minus 7 cells. So doing that, we and other groups have shown that detection of MRD is a very strong prognostic marker, which will help to individualize treatment, because these patients, of course, need additional treatment, whereas those patients not. And so far, most of these patients were treated as one group. So only about 50% of patients with AML have a suitable, highly sensitively detectable marker using, for example, real-time PCR. We now know from NGS analysis that single base pair mutations are present in many patients, and these are very challenging to be detected with high sensitivity using qPCR. So our question was, can we detect such mutations using next-generation sequencing? This is just a scheme illustrating the approach. When you have a standard coverage, probably about 40-fold per amplicon, you can detect this with a sensitivity of probably 5%. But if you increase the read depth per sample to probably 400,000-fold, the sensitivity achievable is much, much higher. So this would have major advantages. It would be easy because you could adapt it to different amplicons very easily, and you would not need very much of validation per amplicon. The disadvantage so far was the price tag, which with the methods available so far, especially the 454 technology, was much too high to be usable in the clinics. But several groups have already shown that it might be an approach to do. So we asked, could we do that on the PGM? We used the Fusion-PCR method for Amplicon resequencing. This is just an illustration how this works. You use PCR with very long sequences to adapt to align or to fuse sequences to the amplicon of choice, which contain either the barcode and also the adapters needed for the library preparation. This is the workflow. Essentially, it is quite standard. Instead, that we start with the Fusion-PCR initially, which is a single-plex unidirectional PCR. You pool then the amplicons, which you want to use on your chip, and you purify that with the, we use the AgentCure beads and measure that to have a sufficiently diluted library stock to be used in the emulsion PCR. This was our first shot with this test, and we were a little bit disappointed to see that there is a high degree of background calls. This is the non-consensus base calls depicted here for the entire amplicon, which was tested here. And you can see that there's a median error of probably 0.25% with a very high variability. We then went on to see whether we could improve that by using a different proofreading enzyme. And indeed, we were able to show that for the entire read length, but also for particular positions, for example, this position we were mostly interested in, you could substantially decrease the background. We then went on to increase also the template amount, because currently the template amount is tweaked to have very low amounts of template to be very effective. But when you want to go for minimal residual disease, you should probably increase the template amount, because a normal human genome has about 6 picograms of DNA. So you have 8,000 genomes within 50 nanograms, but almost 40,000 genomes within 250 nanograms, which is, of course, important to have a high sensitivity. And with more DNA, you could also decrease the number of PCR cycles necessary, which would decrease the background. Combining these two approaches, we were able to substantially decrease the number of background mutations which were seen on specific bases. Here again, that particular mutation in the JAK2 tyrosine kinase. And we then went on to perform dilution experiments, diluting a cell line with the mutation, carrying the mutation in a background, a normal cell line. And you can easily see that there is a very linear correlation, log linear correlation between the spiked alleles and the found alleles, down to at least 0.01%. And if you use the 318 chip and achieve coverages of more than 600,000-fold per read, you could even go down probably 1 in 50,000 reads. And we tested that then also on a clinical scale. We went on to, this is a patient after transplantation carrying a JAK2 mutation in exon 14. And this patient actually was diagnosed with reoccurring recipient cells indicating a relapse of his disease at about 1,000 days after allogeneic transplantation. And we tested the JAK2 mutation in this sample. As I told you, the background which we observed is 0.01%. And we could see that the increase over this cutoff is about 6 months earlier than this method which is currently used in the majority of the patients. So you're gaining a lot of time which could be used for intervention, for example, using donor lymphocytes infusion. So we have now developed that and tested that for a number of additional targets, for example, IDH1 or 2 mutations depicted here. And we can routinely achieve a sensitivity of at least 100,000, sometimes up to 100,000 cells by combining, for example, this strategy with a specific PCR clamping strategy where you block the amplification of the wild-type allele, which is very interesting that you can do that kind of diagnostics on a DNA basis. What could be a next step, a potential next step? Of course, at the moment, costs are still high, not that high than with other platforms, but they are high, around 60 to 70 euro per analysis. But what could be interesting to do that with a coverage of 500,000 reads on a proton instrument, since you can achieve there easily 50 million reads, you can envision that you will be able to achieve costs below 10 euro per sample, and even more with the new releases. And that might be a very interesting alternative. And I'm very thankful to Crescenti Ainali for providing me these data. What we also did, and you have heard about that particular topic several times within this conference already, is the detection of mutations circulating DNA. So you know that tumors are liberating DNA into the circulation, either, for example, by apoptosis or by necrosis, and you can detect then this DNA in plasma, in the plasma, cell-free DNA preparations. And several groups have already shown that you're able to monitor disease using, for example, next-generation sequencing. And we have also done this on our PGM platform for patients, for example, with colorectal cancer carrying mutations of the KRAS gene. And you can very nicely see that a patient without chemotherapy who denied to receive therapy has an increase over time of this mutation load in the plasma, whereas another patient under chemotherapy has a very good response to chemotherapy clinically, and also decrease of his mutation load in the plasma DNA. So summing up, I think we are all sure that DNA alterations will become key lesions in the classification of many tumors, and especially also in AML. I hope that we will be able to generate, and we're quite looking forward to use these dedicated amplicon panels, especially, for example, on the Ion TORRENT instrument, to do more comprehensive analysis of patients and diagnosis. I hope I've also shown you that next-generation sequencing has also a potential to use ultra-deep analysis of known alterations, which will be probably useful to assess alterations with very high sensitivity to monitor, for example, disease or to study clonal evolution. With that, I would like to thank the people in the lab doing this work, especially Seva Herold and Caroline Schuster, who actually did this. The bioinformatic people, these are the most important actually in all these analyses, and especially, of course, Ion TORRENT and the Falcon team for supporting our studies. And with that, I'd like to thank you for your attention.